Today, I'm going to give a science talk about gene therapy. Now, this video is actually adapted from a presentation that I gave at a conference last year, and gene therapy that is something that I've been involved in very heavily in my work the last few years. So first, what is gene therapy? Essentially, gene therapy is a way of treating a disease that has a genetic cause. In other words, there's a spelling error, think of it, in somebody's DNA that is causing their body to have some disease. And there's two basic ways that you can potentially fix that problem. One is to go into a person's own gene and correct the spelling error. The other is to transfer a new correct copy of that gene into their body. Now, all of the gene therapy treatments that are currently approved or in clinical trials use the second approach, which is more specifically gene transfer therapy. And then we'll go into a little bit of the nomenclature of genetic diseases. There's what's called a dominant disease, which means that you have, in general, two copies of every gene. And in a dominant disease, if you have even one copy that's incorrect, then you have a disease. Most diseases are recessive, meaning that if you have one bad copy but one good copy, then you're still healthy. It's only when you have both copies that are bad that you have the disease. And usually that's because you're not able to make some protein. So giving a new copy of the gene which can make the protein will prevent or correct the disease. So for gene transfer therapy, specifically for recessive diseases, we're trying to put a new good copy of the gene into somebody's cells. So one, how do we get it into the cells? Secondly, exactly what is the gene that we put in going to look like? Is it going to look exactly like the copy that a person already has, except without the error? And how do we know that it's actually going to work? So the basic way that all of the gene therapy that is currently being tested or has already been approved uses a virus to put in the gene to a cell. Okay, the particular kind of virus is called an AAV. And it's a kind of virus that actually doesn't make anybody sick. And also, we've taken out the genes of the virus so it can't reproduce itself. Now, normally, viruses reproduce themselves by putting their DNA into a cell and getting the cell to make more copies of the virus. But when we use an AAV for gene therapy, we take out the virus's genes. It can't reproduce itself. Instead, we're using it to deliver the therapeutic gene that a person needs into the cell. So why do we use AAVs? Well, because that's how viruses reproduce. They've gotten very good at putting DNA into cells. So we're taking advantage of that to deliver a new gene. The problem is that our immune system only sees that it's a virus, not that it's actually a good guy that's there to help. So we still have a little bit of a problem with the immune system reacting to the virus. Now, there's a few alternative approaches which aren't as well developed and aren't being tested in clinical trials in people yet. Okay, we've gone over AAV. Now, another disadvantage of an AAV is it's very small, which means that the biggest gene that it can carry isn't that large. Other viruses have a bigger carrying capacity, but they typically cause more of a reaction from a person's immune system. There's also ways that we can put in genes without using viruses, but because viruses are so good at putting DNA into a cell, we really can't do as good a job unless we use a virus. Okay, so what do we put in the cell for the transferred gene? Well, the way that, okay, so first, the basic purpose of a gene is that it's an instruction for your body to make a certain protein. A protein is a chain of amino acids. There's about 20 different ones that our body normally uses. And in each protein that the body makes, the amino acids have to put together in a very specific order, and the gene has the instructions for the ordering of the proteins that are of the amino acids to make that protein. Now, the parts which code for the order of the amino acids are called exons. They're the things that are shown in red in the upper left-hand corner of the figure. But most of the gene isn't actually made of exons. There's other stretches of DNA that do other things. And so what happens when the body makes a protein is it first copies the DNA into another molecule called RNA, and then it splices out all the parts except for the exons. And the introns, those are the non-coding parts of the DNA, are about 98% of most genes. So after you splice together the exons in the final form, the gene is much smaller than it originally was. You can think of the splicing as kind of like filming each scene of a movie, and then after you have it all together, you splice all the scenes together in the final form. So before you do the splicing, the DNA of a gene in our genome is far too big to fit into an AAV. But after the splicing, the compact form of DNA, which is called cDNA, for most genes will fit into an AAV. But there's a couple disadvantages. One is that sometimes for a lot of genes you can have alternative splices of the exon, so you can make different forms of the of the same protein. Now, for after you've already done the splicing in the transferred gene, you can only make one version of the protein, but a lot of times that's good enough. Okay, so we've got the cDNA that has the instructions for putting together the amino acids to make the protein, but we need a couple of other things to be put into the cell for this to work. Okay, one is what's called a promoter, and the reason for a promoter is that it turns out every cell in our body contains all the genetic instructions for every protein that our body ever makes. But not every cell makes every protein. That's actually what determines, what cell is a muscle cell or a skin cell or a blood cell, which proteins they make. So which cells make which proteins and how much is controlled by a promoter. So the promoter, will tell a cell, okay, if you're this type of a cell, then you should make this protein. If you're not, then don't make this protein. And also how much of the protein that it will make. So I'm going to focus in this talk about genes for muscle cells to treat neuromuscular diseases. So this would be, you'd use a promoter which turns the gene that you're putting in on only in muscle cells, sometimes in heart cells. In addition to the genetic instructions for the cDNA and the promoter, you need a few short pieces of DNA to mark the beginning and the end of the gene so that the cell will recognize it as a gene and can transcribe it. Okay, so to summarize, you take the DNA for the naturally occurring gene, splice together all of the exons, put it in a compact form, add the promoter and the other stretches of DNA, put it into an AAV, and then you can, in most cases, just inject that into a person's bloodstream. And the AAVs will put the gene into a variety of cells, not just the cell type. If you're trying to do gene therapy for a muscle disease, it won't only go to the muscle cells, but because the promoter only turns the gene on in the muscle cells, that's not really a problem. Okay, so I'm giving here a list of a number of diseases, genes involved in different neuromuscular diseases that are either being worked on or in clinical trials or in some cases almost approved. And so for most of these genes, with only a couple of exceptions, the cDNA, after splicing everything, all the introns out, are small enough to fit in a single AAV. Now, there's a couple of exceptions, but we are still working on gene therapy for these because there's a couple more tricks that we can play. Okay, so one way is you can divide the gene into two halves, put each half in a different AAV, inject the person with both of the AAVs, and then the two halves of the gene have a little bit of an overlap, which not always, but some of the time, the cell will reassemble the DNA and have the complete gene to make the protein. It's sort of like, going to IKEA and, buying furniture that requires some assembly when you get it back to your house. Another approach is that for some genes, some parts of the gene, of the protein, are more important than other parts. So we can pare down the gene to only its most important parts, fit that into a single AAV, and get not the complete protein, but something that works pretty well. Okay, so that's what we're going to try to put into people. It turns out that this is always tested in animals, usually mice first, to make sure that yes, it can truly deliver the gene and help whatever disease you're trying to treat. But then you need to do clinical trials before it can be approved. So the first thing you want to know is it safe? Can you give this treatment to a person without harming them? That's where AAV is much easier than other forms of gene therapy because it doesn't provoke that much of a reaction from your immune system, unlike other viruses. Secondly, after you deliver the gene, is the person now able to make the protein that they weren't before? Secondly, and this is the hardest part, are you actually making the person's disease better? So you need to, define a few things. What is the natural course of the disease? What tests are you going to do to demonstrate that the person is actually getting better, or at least not getting worse, like they normally would without treatment? And then you always do this in a blinded way, so that neither the person already knows that they've gotten the real treatment or a sham, and neither does the person evaluating them.